Hello and welcome to this webinar titled Extractables and Leachables, Testing Using a Quality Risk Management Approach, hosted by Biopharma Asia magazine and presented by Davao Tapiowala, Principal Scientist at Pfizer, and Satish Kumar Mohanvelu, Life Sciences Management Professional at MilliporeSigma. My name is Stephen Edwards and I'll be your moderator. Now, please allow me to introduce our first presenter. Davao Tapiowala graduated from University of Pune, India, with a BE in Chemical Engineering in 1997. He joined the University of Louisville in 1998 and completed his MS in Chemical Engineering in the year 2000. His research thesis was increasing single-cell protein from distillery waste. He has over 18 years of experience in the biotech industry. He has worked in a validation group during process and equipment validation, engineering group to design, technology transfer and CMC filing of therapeutic, blood plasma and vaccine processes. He has both upstream and downstream experience in microbial and cell culture platforms. In 2016, he joined Pfizer as Principal Technology Scientist to design Brownfield Capital project to technical transfer of commercial vaccine production to Pfizer site. He has vast experiences of design and implementing single-use systems in mainstream manufacturing and actively involved in Bpog. I will now be handing over to our first presenter. Welcome, Dhaval.
Thank you, Stephen. Good morning, good afternoon, and good evening to people across the world. I would like, first and foremost, I would like to thank Biopharma Asia for giving me the opportunity to present. And today's topic is going to be using quality risk principles towards assessing extractable leachables for your single-use systems in the processes. So first of all, I would like to thank all the teams as part of the Bpog. As Steve mentioned, that I'm actively involved in Bpog, and it is a cross-functional, cross-company team that came up with the biggest challenge was how to standardize the extractable and leachables profile assessment across different companies. And then this is basically the two working groups within the E&L sub-teams. One was the Extractable working group, which published a standardized extractable protocol, which has been now widely accepted by also the suppliers in terms of generating the extractable data. And then the second one we came up with was the Bpog's Leachables Best Practices Guideline. And my presentation will be more focused on the risk assessment piece. This is the website of the Bpog, and then these are the live links for the two documents. Well, first one is the Extractables protocol, and the second one is the Leachables guide. So, as I mentioned, my talk is going to be focused on regulatory expectation and end user challenges, and then the risk assessment using a standard model approach, which is also used or adapted by Pfizer and some of the other member companies, or some elements of these approach have been used in terms of the risk assessment. And then the end user perspective of what the Bpog recommendations are. So as everybody is aware that the single-use technology is getting widely accepted in the mainstream production, where it's for the use of the systems. So if you look at the standard process now, more than 100 plus single-use components are used in the process, and in order to qualify them requires a lot of E&L studies with limited or inadequate suppliers' data. So the extractable protocol was mainly driven towards that. Even the regulatory requirement, CFR 211, says that the equipment used in the manufacture of the drug shall be constructed so that the surfaces that contact components in process material or drug product shall not be reactive, additive, or absorptive. So basically, we don't want to have any equipment components adding to the product stream or the process stream. These are typically assessed as part of the process design, during the process design, and as part of the raw material. In the old days, traditional brick and mortar facility, we would have a stainless steel facility where it's proven that it does not add that much, but when we are looking at different components like for single use, we have to worry about the extractables and the propensity of leachables into the process stream. So what is extractables and leachables? Extractables are basically the chemical entities that can be extracted into the process stream in the presence of a solvent or controlled conditions. And those conditions, when they are tested, are more aggressive than your normal operating conditions. And the leachables are basically chemical entities that are come into the process stream for during normal use. So again, when people ask me, I usually use define them in layman's term as extractable is like IOQ, and the leachable is around PQ, which is validating the equipment for your process. So again, in the layman's term, that's how I define extractable leachables. People do use these terms synonymously, but they are two completely different components, or they mean different things. So the regulatory expectation, you can see, as I mentioned, the CFR 211 is very clear. There is E&L data for BLA submission is a must-have based on the past regulatory request from FDA, EMEA, other regulatory agencies, and even Ingrid Markovich from CBER had presented in 2014 in the ASTM workshop, which clearly identified that E&L assessment is a must if you're using single-use technology. And the leachable studies must be incorporated as part of the stability program, which is, again, using the quality risk management principles. So ICH Q9 Quality Risk Assessment, QRM, says that it is very important, gives us a guidance that it's very important to understand that the product quality should be maintained throughout the life cycles, such that the attributes that are important to the CQA of the drug or the product remain consistently with those that are used in the clinical studies. It's very critical that you understand your process right from the clinical. A lot of times, as part of the tech transfer to manufacturing facilities, you may change some of the material, and you then have to do additional studies. So it's very important that we take a science and risk-based approach to understand the propensity of E&L in the final product. So this can be done as the end user develops the processes and performs clinical trials to assess the CQAs for their indication. They develop this knowledge base. This can be documented in the R&D studies, process descriptions, SOPs, technical reports, and that can be used as a guiding document to make the assessment for the extractables. Risk management principles are basically are based on three legs, basically severity of the harm, occurrence, and detection. So taking that forward, we are trying to look at the leaching propensity assessment. So again, as I mentioned, when we try to make these assessment, you can either be smart about it and so use the process understanding and then combine the extractable data, evaluate, and then make the risk assessment for the leaching propensity. If it is acceptable level of risk, then you don't need any additional leaching studies or testing. If it is unacceptable, then you definitely need to do leachable studies. And so how to go about it? The other option is that you test every single-use component. Again, it can be a very expensive affair in terms of trying to get all the studies done in terms of time, resources, and also money. And then ultimate goal is, of course, the regulatory approval. So those are the two options we can use. Test everything or use a risk-based approach for testing only the components that are deemed as high risk. using the QRM principles. So it is a risk assessment, again, not a safety assessment, because this question has been asked that we have to look at the toxicology study. No, it's a risk assessment that based on the process knowledge and also the impact on the CQA, then you can calculate the toxicity. So for this risk assessment, you use available extractable profiles and then calculate the safety profiles wherever possible. A risk assessment, of course, we got to be cognizant that a risk assessment is subjective and depends on the user and the user's appetite for the risk, and as it will become clear as part of the model. Adjust the model to the specific company. Again, this is just a model that provides a guideline to a standardized approach. And as I mentioned earlier, a lot of member companies have been using some elements of it, or even if they are using everything, all the elements of this risk assessment, they have tailored it to their needs within the company. So using leaching propensity assessment as a harmonized practical approach gives you the relevant gives you the flexibility to include the relevant scientific considerations. The leaching propensity, so how do you define leaching propensity? It is the unknown leachables from the polymeric material entering into the process stream and ending up in the FDP at a concentration of concern. It is a likelihood, just to point it out, that it's a likelihood and it's not definite. So again, it is impacted by the component and the process conditions. So the challenges for the end users are both external and internal. One thing is the ultimate goal is the risk evaluation, the patient safety risk, the product impact manufacturing risk, and the regulatory approval risk. So how do you go about this for the end user? So you can use the lean process, useful supplier data, your process knowledge, lessons learned from 483s, which is you typically published, so you can always use that published data, and the science. So you got to be smart about it and see if we can, instead of using the traditional ENL approach, can we use a smarter way, which is a science-based risk assessment. So how do you go about gaining the process knowledge? So the drug development phase is basically starts from discovery phase 1, 2, and 3 before we commercialize it. Again, considering the vaccine model, we also try to do is after phase 2, we pretty much lock down the process. So that's why you see that a long line where the importance of the ENL profile becomes more critical. So what are the considerations that you can take? So you can, as part of the discovery and also phase 1, phase 2, as a process development scientist, you can take into consideration the material of constructions. Definitely need to look at the availability of the material as a class 6, whether it's compliant for tubing. There are other regulations that we need to comply with. And based on that, you build a standard approach of the materials that you want to use throughout the process with the eye for the end goal, hoping that the process and the product is commercialized, and what sort of single-use systems you're going to be using. As we get into phase 2 to phase 3, we should be starting to develop the extractable profile, the risk assessment evaluation, start gathering all the data from the vendor, and start making an assessment, because it may delay your filing if you find that there is an issue with a material that wasn't used as part of the clinical study but now is required to be used in your production facility. And we may have to do additional studies to gather data. So ultimate goal is the regulatory filing. So this is a nice chart that we've created to help people guide what sort of expectations at different phases of the drug development. So the leaching propensity assessment considerations are basically distance along the process stream. So if you are using a particular component in media prep or cell culture, which is so upstream of the process or far away from your drug product, does that have the same severity as your filter that is being used for your final filtration of your drug product? Of course not. So we should always consider the distance along the process stream. The thermodynamic consideration, the solvation power. So if you are the criticality of using a standard buffer versus a solvent is a lot different. So we should always consider the solvation power. The thermodynamic conditions, what sort of temperatures we are using at. Is it at room temperature where the propensity is less versus whether it's freezing? Do we need to factor in the freeze-thaw cycle, etc.? And then the other kinetic considerations are the exposure duration. Longer the longer the material is stored in a single-use component, the higher the risk is of leaching propensity. The exposure temperature, as I mentioned, and then the dilution factor, whether your material is concentrated versus your diluted material, and also the volume to the surface area. And then this is basically just the definition. I'm not going to go through each and every, but as I mentioned in that cartoon in the previous slide, with the things that we need to consider is distance along the process stream, exposure duration, exposure temperature, exposure surface area, exposed solvent volume, dilution factor, and the solvation power. And all these factors are considered into this little chart where we have tried to give a guideline of around what the criticality is going to be. So if it is a distance, as I mentioned, along cell culture upstream, the rating would probably be 1 compared to a 9 when we are dealing with formulation and filling of a drug product. Same thing goes for the exposure of the temperature. So if it is if it is frozen or less than 2 degrees C, it is going to be a less likelihood of leaching propensity. And then as we go greater than 30 degrees C, it will have a much more higher severity and the likelihood of leaching. Exposure time, we have tried to come up with define it, or what is transient. Is it less than 30 seconds, less than a minute? So we try to standardize it as, say, less than an hour, and then short is 1 hour to 1 day, and then medium is in the magnitude of 1 day to 1 week, and then long is going to be when you do a long-term storage. And so forth. So the solvation power, if it's whiffy, it's going to be very unlikely that you're going to be leaching out anything. But if you're dealing with a solvent, say it's ethanol or polysorbate or something, then the propensity is much, much higher. And the dilution factor, if you are using, say, a buffer, 500-liter buffer in a 500-liter tote, the dilution factor is going to be very large. So the ranking would be 1, and if you are going to use a 20-liter or a 10-liter concentrated product in a bag, the dilution factor drops significantly, and then the criticality ranking goes up. So what do we do with all these risk assessments? How do we come up with more of a standardized format? So using all those factors, we came up with some sort of a leaching propensity ranking. So where we use all those factors and we have assigned the weightage to those factors. The distance along the process stream we've used as 40%, and the remaining are around 15%. Again, this is just a guideline. We have used calculation also in Pfizer. And then based on that, we have used we calculate the risk assessment. So if it is from calculation is 1 to 3.6, then it's low. 3.7 to 6.2 is medium, and 6.3 to 9 is high. Again, I just want to make it clear to everybody is that this is just an example So if it needs to be tailored to your needs, it can be tailored to your needs depending on the risk appetite and also your process needs. You can change your weightage on these different considerations. So just as an example, we use this as an example to define the process from one of the end user company. So if you look at these, the upstream was defined, and then purification, and then formulation piece. And as you walk through the process, you can see that the upstream will probably have a less criticality depending on the distance from the process stream. So taking that, defining that process and seeing where we are using this different single-use equipment, we came up with a profile, a working example of what these rankings should be and what the risk levels are. And if you can see, we've clearly been able to define that as low, medium, high. And if you were to use the old traditional model, a lot of these single-use elements would have to be tested. But based on the risk assessment, we are able to have only one high, and then the medium is also, it probably falls in the gray area, but the medium can be defined as whether it needs to be tested or not based on the end user's risk appetite. So it just gives you a little bit more granularity in terms of what can be tested and be smart about it. It can be and minimize your costs and also the time and effort. So one of the things was that how does this model compare to the end users for the companies that are already using this risk assessment? And if you can see, we did get a response from 9 different companies, end user companies, and the model said that the initial observation was that it is very close to the current industry practices with a very, very small bias. So it's good to see that the LPA, Leaching Propensity Assessment, is very comparable to the current practices within the industry. And so how do we apply that? So as we go through the different phases of the drug development, we identify the risk, assess it, especially when we go through the extractable assessment, and then we mitigate those if we find that the safety toxicology studies are important to the risk of the patient, then we need to mitigate that. And we communicate back and also go through that cycle. So this is an effective approach, ensures a high quality of drug product to patients. It's a proactive tool. As I mentioned, we develop the knowledge base for the process, improve the decision making, and then as part of the life cycle of the product, we need to continuously assess if anything changes from the supplier notification. Then we have to go back and reassess the risk assessment. So it's a very good tool to ensure that you are able to maintain high quality of drug to the patient. So this is a very busy chart, but if you go to the leaching, it does talk about the process flow. So when we are looking at the process, we should be taking into account all the raw materials and list them. So the key would be basically that the key would be that we are looking at the user level of the list of all the materials used, list of the process aspects, and then once we make that material construction, etc., and then we look at the extractable profile. So first thing we need to answer is that is part of the polymeric material. If it is yes, then we need to assess whether it's a process stream or not. If it is not a polymeric material, or if it's not a process upstream contact, then you don't need to do further E&L assessment, and that can be taken out of the assessment spreadsheet. Then you got to evaluate the E&L risk assessment based on the extractable data, and also consider all the 6 different components that we were considering: the exposure duration, the exposure fluid, the dilution factor, the volume, and the distance of the process stream, and assess the leaching propensity. If it is medium or high, you can do additional studies. If the risk is low, then or acceptable, then no further E&L or leaching studies are needed to be done. And as we go through that and answer the process flow, if the leaching studies have to be done, then if the risk is high, either you can mitigate the risk by changing the component and going through that risk assessment again, or you can perform a leaching study, and that would be also included as part of your filing studies. So that's what the quick process flow for doing a risk assessment. And then based on the calculation as I showed, we can break down the risk into low, medium, and high. Low is typically the minimum requirements are class 6 and compendial data. Low requirements plus supplier extractable data assessment if it is medium, so to demonstrate that why it's medium. And then if it's a high, you require all the requirements from the medium plus leaching studies. So to make sure that we have demonstrated that the leaching propensity in its all likelihood is not impacting the safety of the patient. So the conclusion is the Bpog team has developed a practical and a flexible approach to assess, because the biggest question at that time was everybody agreed that we have to do an E&L risk assessment. The question was how we can standardize it. The other question was also how we can demonstrate that we can standardize it across the company. So if we can come up with a method, then all the regulatory agencies are also able to look at these models in the same way. And then, as I mentioned, the biggest challenge was also to standardize the extractables data for the vendors that are supplying to the end users. So all the end users can also look at the data in a standard format. So one of the things that has been accepted now is basically the vendors are following the Bpog protocol for extractables so they can generate the data in a standard format. And then again, as I mentioned, that this is just a best practice guideline, so we wanted to also bring in enough flexibility for the end users that they can adjust the parameters based on their process needs and the company requirement. So, and as I mentioned, Pfizer has, at least on my site, we have been using some elements of these standardized approach from the Bpog leachable studies. Any questions?
Thank you, Dhaval. But once again, I'd like to remind our viewers that there will be a question and answer session at the end of the webinar. But please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second speaker. Satish Kumar Mohanvelu is a marketing manager at MilliporeSigma, USA. In this role, Satish is responsible for implementing the Improve program with a specific focus on filtration and single-use portfolio of products. Through this program, very comprehensive dossiers are created for products to support customers globally with material qualification, risk assessment, and process optimization initiatives. Extractables information is one of the key contents of the Improve dossiers. Satish is a PMP and a postgraduate in biochemistry with over 18 years of experience in the biopharmaceutical industry. Having spent 10+ years with the organization, he has held various roles in supporting and promoting the validation services in the past. In this capacity, he directly managed the filter validation services and laboratories in India successfully for many years. I will now be handing over to our second presenter. Welcome, Satish.
Thank you, Stefan. I would also like to thank Dhaval for nicely presenting the concept of risk management and in relation to the Leachables best practice guide as per Bpog. So I would like to discuss further many of the concepts that I'm going to talk in my presentation, which is titled as the risk-based approach towards extractable studies for single-use systems used in biomanufacturing processes, would have a certain overlap, but many of the concepts may be presented in a slightly different way, but Dhaval's explanation actually gives a nice segue into what I am going to discuss in the next few minutes. As we discuss this topic, we would begin with a quick analysis of how the single-use systems have influenced the biomanufacturing processes today. From there, we would get into an overview of three other topics. Risk-based approach of extractables and leachables. Here, I would again be touching upon a few sections of the Leachables best practice guide by Bpog, and then focus briefly on extractable studies, available methods, and approaches when it's applicable to the single-use components. Then I will conclude with finally on the risk mitigation strategy, more an overview on more standard approaches that have been followed across. Single-use adoption has enabled desired process efficiency improvements in biomanufacturing, both at clinical and commercial scale. In fact, today, at the clinical scale, nearly 85% of process operational steps are handled through single-use systems, and there is good data to support that. With a very good market and a steady growth over years, single-use systems have contributed to an increase in efficiency and productivity with an overall improvement in the biomanufacturing performance. I've been following the bioplan reports over the years and fully agree that single-use systems reduces capital investment, cleaning efforts and associated validation processes, and the risk of cross-contamination that could come otherwise. Despite of the several advantages, there are a few lingering concerns on complete adoption of the single-use systems. And one among them is the availability of the extractables data and the potential concern of leachables entering into the process stream. Today, most of the unit operations in a typical biomanufacturing process consist of polymeric compounds, polymeric components. For example, if you look at the monoclonal antibody platform, which I have illustrated here, you can have plug-and-play modular unit operations at any of these steps as described. Similarly, chimeric antigen antibody receptor T-cell processes are mainly on a single-use platform. There are regulatory guidelines and regulations for extractables, leachables from the US FDA, EMA, and ICH. Dhaval already touched upon this topic in his talk and presented the details well. I would like to specifically point to a CBER recommendation as said by one of the senior CBER regulatory officers. This might have been seen in several presentations in the past, but I'm still referring to this because of the clarity that this message conveys when it comes to risk-based approach. This is a dichotomous approach. When we look at the risk, we have to consider multiple factors into account. If there is no relevant risk associated with the materials in question, vendor's data can be cross-referenced with a detailed justification. If there is a relevant risk, determine the toxicity based on maximum dosage. If this yet presents a safety risk, perform a Leachables evaluation and testing, and if the risk assessment could affect the product quality, conduct studies to assess the impact on the same for the products. Please remember what we discussed over here, because many of these comments or points would be very well seen in the further slides that I'm going to discuss. Quickly pointing to the ICH-Q9, the risk evaluation process should be based on scientific rationale and ultimately link to the protection of the patient. It clearly highlights that higher risk requires higher levels of qualification. Any level of effort or formality in the form of documentation for the quality risk management processes should be well commensurate with the level of risk in the process. This is a good prelude for us to enter into the next topic, which is on a risk-based approach for extractables and leachables. As also Dhaval addressed, it is the biopharmaceuticals manufacturer's responsibility to qualify various materials used in the manufacture of a biological drug so that they meet specifications for toxicological risk, process performance, and product quality. When it comes to extractables and leachables, every manufacturer adopts a risk-based approach. The approach could vary. Let us look at one of the decision trees as given in this slide. It is important to note that a risk-based approach does not necessarily require that extractables and leachables be performed for every single-use component used in the process. The approach as illustrated here can be looked at in 3 different steps. The Bpog's Extractables and Leachables Best Practices Guide could again be used as a standard reference when building over these approaches. And the risk propensity assessment is again a very useful tool in putting together a strategy. The risk assessment could look at one unit operation at a time or the overall process. At the pre-work stage, it lets you to set an assessment boundary around your strategy. The parameters to do the risk assessment must include, but may not be limited to, the 6 different steps or factors that has been identified in this slide. Some organizations also set a cutoff score to make a decision on yes-no criteria when it comes to risk that could be evident or obvious when dealing with these factors. Whenever there is no relevant risk, the vendor data can be utilized to support the justification. That is something which we understood even in the previous presentation, as well as in the regulatory reference that I made earlier. It is up to the organizations to decide whether or not to perform an extractables risk assessment at early phases prior to phase 3. Similarly, leachables evaluations is usually not required for materials in manufactured during early phases of clinical trial drug development. With an increase in surface area, it provides a more opportunity for leachables. Lower process volumes again concentrate the leachables in the process and represent a higher risk. Any pretreatments such as autoclaving or gamma irradiation or WFI flush may again affect the leachables profile. The step 2, we would look at the extractables study design. At this stage, we are sure that we need the extractables data to support your assessment of the process or a product. The most preferred approach today is to have extractables data as per the Bpog protocol. At this stage, using the data, the safety risk based on maximum dosage of potential leachables from the extractables data would be assessed. The third step would be to decide on a leachables study requirement and a related safety assessment. If the assessment presents necessarily a safety risk, that would be a trigger for a leachables study. Any leachables present in a leachables study should be identified, quantified, and its toxicity assessed as a part of the safety assessment process. So the risk assessment and the safety assessment are slightly different processes, but much related, and both have to go hand in hand when it comes to assuring patient safety. Let us illustrate this risk-based approach through a typical example presented in this slide. Again, this refers to the Bpog's E&L Best Practices Guide, and many of these terms have been defined during the previous presentation. Consider any typical biomanufacturing process. The influencing parameters are listed in the first column. And the column 2 lists the risk rating in a serial order of each of the factors. And the typical production conditions are the product and process characteristics is covered in the mid of the table. And the last column indicates the related weightage. It can be noted that the higher weights have been assigned to final fill operations due to the greater risk of impurities in the final drug product when we look at the distance along the process stream. You may be able to arrive at a leachables risk ratio by simply multiplying the risk ratings about the weightage for the components in a particular step or of the process, and by adding such information for all the components of the assembly. The risk score could be set to classify the assessment as a low, medium, or high status. As defined by the Bpog Risk Assessment Guide, a lower risk component should meet compendia requirements such as USP Class 6 information, while a medium risk requires an elaborate extractables data and a higher risk requires leachables profile along with the extractables data. Now, the classification of lower, medium, and high risk has already been discussed. The same information can be seen in this slide on how it impacts a typical process flow. Again, illustrating the risk categorization along a typical process. The bags, single-use bags used at the stages of drug substance or drug product used for transferring or mixing applications present a higher risk than any other components, as you can see here pointed as connectors 1, 2, and 3, where very low surface area to volume ratio is applicable, and these connectors probably also have lower contact time, and therefore they present a lower risk scenario. So to summarize the importance of having extractables data for single-use components which influence the leachable studies, I would like to highlight 3 important points here. The various regulatory guidelines require that all polymeric materials used in either product storage or drug delivery systems are screened for potential extractables. Higher quality extractables data can guide and define the depth of a leachable study design or the need for a leachable study. Extractables data sets facilitate drug product specific patient safety risk assessment of the extracted compounds. Taking a quick look into USP 665, which is still at its draft stage. This chapter, titled as Polymeric Components and Systems Used in the Manufacturing of Pharmaceutical and Biopharmaceutical Drug Products, also discusses or addresses risk similar to the understanding we gained in the previous slides where we referred to the Bpog approach. The risk evaluation matrix that the draft document presents considers 4 different dimensions that address the risk that a polymeric component will be reached to such an extent that an extractable may be impactful. These dimensions include the temperature of contact or material of construction, chemical nature of the process stream, or duration of contact. The draft is still under review, and we are still waiting for the next draft released for comments. Moving on to the extractables approach for single-use components, I would like to quickly touch upon the IMPROVE program. Our organization presents the IMPROVE program as a strategic way to share with our customers and end users extensive and thorough documentation to support various operational needs such as material qualification or quality risk assessment and process optimization. In this initiative, we present 3 different dossiers. The first one is called as the Material Qualification Dossier. This provides a lot of useful product-based, product-level information such as manufacturing process flow, product characterization and qualification, followed by useful regulatory information. The second dossier that we provide is known as the Quality Management Dossier. This covers information relevant to manufacturing sites, the self-assessment, what we have done for our facilities in comparison to the existing implemented quality management systems, useful supplier and CMO information, and shelf life and packaging validation data for the single-use components. The third one is the Operational Excellence Dossiers, and this presents the complete information on the extractables profile for the single-use components. Now, these 3 different dossiers are also available for raw starting materials and excipients that are provided by our organization. All these dossiers can be accessed and available online through and in their most current version by subscribing to a tool called IMPROVE Suite. Next, I would move on to a discussion with reference to the 2 different methods that are available to handle extractables testing. This is a busy slide. My intention is not to get into the details, but I would like to point over here that when Bpog protocol when it is compared to the USP methods for extractables profiling. The Bpog protocol has 6 different solvents to cover the range of polarity and solubility of extractables at multiple time points using many orthogonal analytical techniques. The USP approach has 3 different solvents, 1 time point. As USP 665 standard is still in draft, in order to prepare for compliance, we have been proactive to include the scope of the method as evidenced in USP 665 in our regular testing approach, which currently is on the basis of the Bpog protocol. And in the future dossiers that we will be releasing soon will have information supporting even the needs of the USP 665. Since we are now discussing the extractables from single-use components, this slide presents the strategy that is adopted by our organization as well as could be similar in most of the supplier organizations. As a supplier, the first step would be to identify all the different components which will be potentially in the fluid path for the product. At this stage, we may be able to assess risk due to the contact with the components, such as like what is the total surface area that is exposed, what could be the contact time, again referring back to the risk assessment strategies that we saw earlier. The next step would be to group all these components, our component information, such as material of construction or what type of resin it is made up of, what is the manufacturing process adopted in manufacturing these components, and then the following step would be to agree on a group representative. Once we decide on grouping these components, the next step would be to pick up one of these components as a representative of the group. And such a representative component should be worst case selection. And then multiple lots of such a component would be subjected for the corresponding pre-treatments and then analyzed as per the methods discussed in the previous slide. Another discussion point around component extractables test strategy would also be a reference to product grouping or product family approach. Number of extractable studies required may be reduced by having an approach of grouping similar filtration or single-use products in a family. These studies are expensive, and unless right strategy is set in, and unless it is based on a scientific rationale, either it could be laborious and intensive, or it could go out of the necessary scope of the end users. When products are present in different sizes with same material of construction, they can again be grouped together. For example, the filters, or even for the single-use bags that are of the same material of construction but are of different sizes. A grouping approach can also involve selecting worst-case representatives for extractable testing. For example, when we are dealing with a capsule and a cartridge, a capsule can be considered as a worst case. When dealing with multiple pore sizes in filters of the same material of construction, a tighter pore size could be considered as a worst case. As we can see in the below illustration over here, which shows a comparison between a Durapore point, which is a PVDF membrane, having a 0.22 micron versus a 0.45 micron. Both of these have same material of construction. Their extractables quantities are very comparable. Once all the extractables testing is done and the data is generated, Bpog protocol has set some of its own expectations in terms of how this data should be presented so that it's useful to an end user for their risk assessment. We present this extractables data through our operational excellence dossiers, which is a systematic compilation presenting sample selection rationale, worst case scenarios adopted, and the extraction methods and analytical approach that is adopted. The results are then presented as an executive summary of detailed extractables profile, which also includes the elemental impurities data as per the ICH Q3D requirements. Going further from the risk assessment, risk-based approach, here I would like to discuss about the risk mitigation as an overview. What we need to understand from this slide is if extractables data is available from the supplier, the data can be used for evaluation, and that's a possibility. The potential leachables that a patient may be exposed to can be calculated based on the process conditions in which the various components are used, product contact surface, production batch size, or drug product maximum daily dosage. This is nicely illustrated below in a stepwise manner in this slide. We typically begin with the analytical data with such information as extractables per unit surface area and additional data of surface area to volume and dilution factors as applicable. Once this data is available, it can be utilized to determine the potential extractables levels in the process at normal operating volume. From a process-based approach, we can further utilize this data to move it to deduce extractables levels in a drug product under minimum batch size conditions, setting it as a worst case. Once this step is through, the individual extractables data that we have can be used to correlate to the maximum daily dosage and other posology, including the route of administration, the frequency of administration, and the total duration of contact. And this could be related to the potential daily exposures, or the PDE. The PDEs are normally represented in microgram per day. It is useful also to convert information from the PDE into concentrations of maximum potential leachables intake to evaluate the impact of potential toxicity. So going over the information that we learned today in the last 1 hour, what we understand is it is clear that availability of extractables data alone would not solve the problem. It is important to actually understand how to utilize this extractables data in deducing a total leachables extractables strategy, ultimately leading to patient safety assessment. In our organization, the MPROVE program and the BioReliance validation services, through these groups, we combine efforts to provide support beyond just the extractables data. The team of experts guide customers with the application of this information as presented in the Operational Excellence Dossier. If required, generate product and process-specific extractables reports, and to design and execute customized extractables and leachables studies for products sold by us used in our customer processes. Apart from this, we also do regular or routine validation services for sterilizing filters and We also have expertise in supporting the toxicological and risk assessment of products and recommendations for risk mitigation steps. So finally, the key messages and considerations out of our discussion today is regulatory requirements evolve and the industry guidances parallelly evolve. So this is a constant changing process. The risk-based approach with a sound scientific rationale should be basically driving extractables and leachable strategies. The availability of extractables data drives effective adoption of a single-use technology in a process. Regulatory expectations require patient safety evaluation and the data to support them. Risk assessment and safety assessment play a very important role in this process. It is important to have an expert's guidance to navigate through this complex subject and assure compliance and drug safety. So that's what I have. I would like to thank some of my colleagues, Jessica Sheshani and Manjula, who have been very helpful to me in preparing this presentation today. And you may contact me if you have any specific questions later, even through my email details have been provided here, and I would be open for questions. Over to you, Stefan.
Thank you, Satish. Now we will begin the question and answer segment of the webinar, but once again, I'd like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now, onto the questions. Our first question is, how is the Bpog protocol aligned with the USP standard?
Yeah, so Stefan, I can take this question.
Mm-hmm.
So when we look at the Bpog protocol, as I presented in my slides, the Bpog protocol is a more worst case approach. It has 6 different model solvents and multiple time points with a detailed analytical strategy. Now, when we compare this to the USP 665 approach, the method addresses, or the method asks for 3 different model solvents. There is a model solvent which is common between both these approaches, which is 50% ethanol. And both these approach recommend a solvent being used at the low and high pH conditions. The Bpog's solvent on the alkaline side or on the caustic side could be a little more worst case because it's 0.5 normal sodium hydroxide, and while in case of the USP, it's more focused around the pH 10. So other than that, as I have the understanding of the latest draft for USP 665, we see that even though USP sets the 3 solvents as minimum expectation, but based on the scientific rationale and justification, additional solvents can be used. So overall, I see that there is a certain amount of alignment, even though the Bpog process is a more robust and a worst case approach, but from a extractables data generation point of view, there is a certain amount of alignment, but we need to wait to see the new draft that comes out from USP 665 to see how they are looking at some of the comments that were shared in the previous comments phase for the draft.
Yeah, I'd like to add to that, Dhaval. So yeah, as Satish mentioned, that the USP 665 is currently still in draft, and the Bpog protocol is much more robust, and it's based on the scientific considerations as well as the use of the solvent in the biopharma industry. So a striking example is the 0.5 normal hydroxide, which is a high pH alkaline pH solvent at 13, which is typically used as a cleaning agent. So that's one of the reasons why it is at pH 13 instead of pH 10. But there is a fair amount of alignment, and we have to wait and see how the new draft, all the comments that the industry peer reviews have done are incorporated.
Thank you. Our next question is, are vendors following Bpog's Extractables protocols to generate data?
Yeah, I can take it up from a supplier perspective. At least in our organization, we have fully adopted the Bpog protocol for generating the extractables data. It's been our strategy to look at our single-use components on supporting them with this information. Even though the component library could be huge, many of those products are our own, while there are components which comes from a different suppliers. But overall, the goal is to support the extractables information aligned with Bpog protocol. But we have also taken into consideration of the USB 665 since it is under 1,000, and when this chapter gets implemented in the next years, this could become a more mandatory requirement. So we have also adopted the requirements from there in our testing strategy. I believe other vendors could be on the same path, but in general, the Bpog protocol has been a very well accepted approach today, both among the end users as well as the suppliers.
So yeah, I concur with Satish that the Bpog protocol has been widely accepted across the vendors. From the end user's point of view, I can say that is a general expectation for the end users that the suppliers are able to provide extractable data in a standardized format following the Bpog protocol.
Thank you. Our next question is, in your experience, what components, such as filters and bags, contribute most to the ELs? Is there any general guide on which components in a typical bioprocess are the major contributors?
So, I can take this. Again, referring to the risk assessment that we discussed through this presentation today, it's basically when we deal with components in a single-use assembly, it's important to understand the risk that they pose based on the total surface area, the contact duration, where this component is exactly located in the distance along the process stream. So several of these factors could actually relate to the risk levels that they have. Now, when you look at some of those components, such as a filtration filter or a single-use bag or tubing, the surface area could be quite high, and obviously these present a very high risk, and there is a need for in-depth extractables information on these to do the risk assessment. But again, there could be components which are on a lower risk side, low contact surface area, but this again depends upon each organization, how they look at these components, and in the process, how does these risk assessment strategies bring out an outcome on looking at their impact in the process. So I would say there are certain obvious information for certain components because of their evident exposure and their placement in the process, but there are other components which are actually debatable and assessable. Many organizations set a risk score, like a polymeric risk score. So if a component falls below the risk score, the data that is looked forward could be lesser or more flexible, but when it goes beyond or above the risk score, there is a normal need or urgency for the Bpog data.
So from the end user point, it always depends on what your process streams and the process solvents are used. So because there is no magic bullet that this particular component is bad and this one is good, and it works with all the solvents. So it has to be assessed based on your process needs and the process knowledge, the parameters as Satish mentioned, and it has to be evaluated. One of the key starting points would be material of construction and the chemical compatibility that can be used as a starting point against your process needs.
Our next question. Do you have a more precise date for USP 665 final publication, and will this be mandatory?
So I can take this one. Yes, it will become mandatory because USP, it will become a law and all the end users will be expected to adhere to it, and in turn, they'll be requiring to have all the data from the suppliers. So it will become mandatory. The date is subjective at this point. There's, as Satish mentioned, that we're still waiting for the second draft to come out after the peer review was done. And we have to see what comments are incorporated and what the draft looks like. But it may be within a year or a little bit 2 years. That remains to be seen.
Thank you for that. Next question is, how would you perform a risk assessment in an assembly of components? Will you combine the data of the individual components?
So I can take this one. So the individual, so there's a classic debate, the two different train of thought. So you do a risk assessment based on individual components, or you can do a risk assessment based on the components that are part of an assembly. And it depends on how the company wants to use it. You can easily break it down to each individual component. It becomes pretty lengthy. So you may want to, if those components are not used anywhere else and just with that assembly, then you can take the approach of risk assessing the extractable data for that component. So for example, a bag involves the film of the bag, the tubing that are associated with it, the connectors that go on the tubing or on the bags, et cetera, et cetera. So if you want to break it down, it just becomes a very cumbersome effort, and a lot of times you may not have the data from a small component vendor. So as the bag assembly supplier, it is the responsibility of the bag supplier to ensure that the exhaustive extractable profile has been created for that particular bag assembly. If the individual end user wants to go back and look at individual components, they're more than welcome to do it, and they'll have to probably negotiate with the individual component vendor. So we deal with a lot of times where we are using a particular component and a connector or something elsewhere in the process, and we have to risk assess the extractable profile for that particular component. But if it is just used on the bag or a filter, then it will be assessed as an assembly in general.
We have time for one more question, and that shall be, what level cutoff is Leachables testing recommended? A risk score of greater than 5?
So as I mentioned in my profile, in my presentation, and if you want, there is a link for the Leachable best practice guideline that does talk about the Leachable testing. So we have generated a low, medium, high, and I believe right now the way it is defined as, I'm just going back to my presentation. Give me a minute. Okay, so the low has been defined as 1 to 3.6. The score of 3.6 or lower is defined as low, and then a score of LPA score of 6.3 or higher is defined as high. So there is no black and white that, hey, above 5 is going to be, we need a leaching study. Again, as I mentioned, the gray area is when the LPA score is between 3.7 to 6.2, which is deemed as medium, and again, it is based in order to give maximum flexibility to the end users. That is up to the individual company if they want to do additional leaching studies or not. So it is up to the individual company in terms of how they want to interpret the medium as such. There has been some more discussion around this medium score, and we're evaluating whether we need to just have two high and a low LPA scores from the risk assessment point of view, but it's still a work in progress. I don't know if I answered your question.
Well, thank you. And with that, we will conclude the question and answer segment. I want to thank you all for your questions. But before I finish the webinar, I would like to ask our presenters if they have any closing remarks. Dhaval?
So I'd like to thank Biopharma Asia for giving us the opportunity, and the presentation has the links to the Extractable protocol and also to the Leachables guidelines. Those are very good informative documents that you can use, and some of the questions that were asked you'll find answers in more details. And it does capture also has a chapter for analytical methods and for leachable study design in that Leachables guidance document.
And Satish, do you have any closing remarks?
I would like to again thank Biopharma Asia for providing this opportunity to talk on the risk assessment of extractables and leachables. I think as Dhaval referred, those links in the documents actually provide a lot of understanding on the approach and also to build your own strategies when it comes to risk-based approach. So please take a look into it. And I think there were a few more questions since we have run out of time, which probably we have not been able to answer. If possible, we will try to reach out and share our responses to it. But once again, thank you very much for an overwhelming attendance, and that's from my side.
Well, I'd like to take this opportunity to thank our presenters, Davao Tapiowala and Satish Kumar Mohanvelu, for sharing their knowledge with us. And you can view this webinar and other webinars on demand by visiting biopharma-asia.com/on-demand webinars. Thank you, everyone. Have a great day.